Ça démarre.
Ah, d'accord.
All right. Thank you, everyone, for joining. Tonight, we are very happy to present our very own Frostnip. He'll be presenting his lecture on computational neuroscience. There will be a Q&A session after. Just for everyone's awareness, the YouTube live stream is currently active, so something to keep in mind. But I would like everyone to welcome Frostnip to the stage. Frostnip, it's all yours.
Hello. We could have the first slide. Yeah. So I'm Frostnip, and I study computational neuroscience. There we go. Yeah. All right. So, yeah, that's the cat that's gonna come up later in the presentation. So, about me. I by trade I program computers. I'm good with computer algorithms. That's what I studied in college, and I got really into machine learning and deep learning and backpropagation. It was a new and exciting technology when I was in college, and I was very excited to make the robots, like, you know, from science fiction, you know, that walk on two legs like people do. I grew up reading, you know, Asimov and stuff like that. But then after spending about a year studying that, couldn't really see how that was going to work. And, you know, for what it's worth, nowadays we have robots with two legs and they still struggle to balance on two legs. But then I discovered neuroscience and I decided to study that. And there were sort of two things that really got me into it. One was how advanced neuroscience is. If the last time you looked at neuroscience was in high school and they told you that the brain is a mystery, we've come a long way since then. And also there's algorithms to look at in the brain, and that's what I'm good at. That's my jam. So next slide, please. It's a mystery. Here we go. The neuroscience renaissance. Yeah, neuroscience has really come a long way in the last like 10 to 20 years. And all of the advances in neuroscience have been enabled by other areas of science. It's very hard to know what advance in one area of science is going to help some other area of science and engineering. It's really hard to predict that kind of thing. So like you have better microscopes that are invented, and that helps neuroscience. And all the chemistry that we have, all of the gene editing technology. We know a ton about biology. We have really good robots and computers as well. The robots are really important for running experiments on our behalf, because a lot of these experiments are incredibly repetitive, as we'll see. And then obviously computers, very important. Next slide, please. Yeah. So this is where those robots are going to come in. We take a brain, we put it through a deli slicer to make it into very thin sheets. And then we scan each of those sheets with a microscope, and we reconstruct what it looks like in 3 dimensions. We get a 3-dimensional model of what's going on inside the brain by slicing it up very thinly. And the next slide should have some pictures of that. Oh yeah, those are pictures of neurons in a mouse brain, by the way. Yeah, so this here is a dendrite. And you can see that's the shaft of the dendrite, and you've got these things sticking out of it. Those are the synaptic spines. There's synapses coming off of it. And look at the layers. You can see the layering on this image where they chopped it up. And the thing to notice here is that the thing we're studying is big compared to the layers and the resolution of the imagery. We have the resolution to chop up a brain and see what it looks like inside. We have like microscopes that can do that. For reference, the scale here is the dendrites probably like 10 microns wide, give or take, and the synapses are like a micron in diameter, around about. So these are very thin layers. Yeah. Next slide. Should be a bunch of pictures of, there we go. Neurons. Yeah, there's going to be a lot of pictures of neurons in this presentation. These are from a website called neuromorpho.org, and anyone can download the files for it. So I've actually run experiments with all of these models, not all of them, but mostly just the big, pretty pyramidal neurons. This one I probably have run experiments with because it looks pretty and it was on the front page of their website. You can tell that it's a pyramidal neuron because the apical dendrite is labeled and it points up. That's like saying a map has the north always points up on a map. Yeah, so the data's all arranged, sorted, they're pointing in the right direction, they're annotated, and this is like publicly accessible data. We have a ton of publicly accessible neuroscience data. And on the next slide, I think there's going to be even more pictures of neurons. Next slide, please. Yeah, okay. Oh, there's a mouse. Okay, that went away. Sorry, that was in the screenshot. Yeah. So this is a lot of pictures of neurons. This is a big reconstruction of a mouse cortex. I believe this is a mouse's motor cortex. So this is what makes a mouse's like whiskers move around and its feet move. And it's all reconstructed. You can see the layering here. You can see like the bottom layer looks different than the top layer, not just in color, but the texture looks different. That's because the neurons in those places are shaped differently. They're all that's important. I know it sounds obvious to say that they're growing in different places and they have different shapes, but shapes and the structures determine like where the connections are able to form, because they can only connect if they grow nearby each other. So this is sort of, once you understand this, you can start forming a wiring diagram of the brain and which layers are connected to which and how. And so this is a map of the cortex here. And if we go to the next image, oh, yeah, this also isn't a lot of neurons. There's a lot of neurons not shown, because if you show every neuron, then the whole image shows up as an opaque solid wall of neurons, and you can't actually see the depth of it. Like, you wouldn't be able to see, like, a strand of spaghetti in a bowl of spaghetti. So what you need to do is you need to find a dye that only stains some of the neurons and not others. And this was what they did way back in the day, and they would randomly stain some neurons with a color, and then they would draw them under a microscope. These are some of the original images of neurons. This is what people used to do. Neuroscience used to be a black box, a mystery, because the data that we collected was just so hard to analyze and so hard to collect. I mean, someone had to learn how to draw to take these images. Cadrel did that back in like the 1800s. He's famous in the field. Anyways, so on to the next slide. We've come a long way since that. They're very pretty images. You can still see the same layering structures and oh, here we go. It's a meme. Yeah. These things, they look pretty, and that is important if you want to get your stuff published and people to pay attention to it. And three-dimensional brain images are particularly persuasive. We can move on to the next image. So, yeah. Those are real studies. I checked. Here we have some three-dimensional brain images. These are, again, mouse cortex, I believe. These are false color. It's only showing a handful of neurons, because if we showed them all, you wouldn't be able to see anything going on. False color, I believe these are voltages. So the membrane potential across the surface of the neuron. Neurons have electric pulses that they send out to each other, and this is how they communicate. And so we're kind of seeing that in action here, where some of them are firing and some aren't. And yeah. Next image. Yeah. When they collected these images, the guy who collected them just sort of collected a lot of data without a hypothesis. They got a whole bunch of money, and they're like, instead of testing one person's hypothesis, they just sort of scanned the whole brain of a mouse and published the data for everyone. And one of the things that came out of that was catalogs of neurons. And what you do with these catalogs, you get a neuron, you have the shape of it, and a bunch of other properties of it that you can tell by looking at it under a microscope. And you categorize it based on all the properties that you can measure. You measure like just as many different properties as you can think to measure, and then you put that through a clustering algorithm. If you're familiar with unbiased clustering algorithms, it just looks for groups of neurons with similar properties, and it outputs the categories and all the neurons in those categories. And what do you know? We have more categories of neurons than we thought we did before. So yeah, catalogs, tons of data. We're categorizing it. We're organizing it all. And yeah, next slide, please. And they're all very pretty. Sorry. So, oh, here we go. I'm going to nerd out about this picture for a bit. So this is a mouse cortex. I believe the colorations are... Oh, I don't know what the colorations here mean. It's kind of hard to say. It could just be false color based on the layer of the soma. But anyways, there's a bunch of neurons here. This is clearly a slice of a cortex. The top of the cortex is over here. We see the long dendrites going up through the layers, like there's a cell body here, and we can see that it sends like that shaft all the way up to the top there. Yeah, so like this is the structure of the brain and where everything's connected. This is the wiring diagram. This is a really pretty picture of it. This is a mouse cortex, I happen to know. We can move on to the next slide. Yeah. All the different areas in it. So what we're looking at in this is like an ice core. Like if you had your brain and you cut like a little circular hole and you just sort of cut like a core sample out of your cortex, it would have looked like that sort of column. So the top of the brain is the surface. Even though your brain is round and there's sort of parts that are up underneath, under your chin, those are considered the top of the brain because it's the surface. It's not really the top, but you know, the surface is, you know, can always put at the top of the image, just sort of for sanity. And that's sort of what we're looking at. And what we're looking at here is yes, science. We're looking at how they they got the electrical properties of each of those categories of neuron that we were looking at in the catalog. So you take each like stereotypical category and you find the chemical properties with different experiments and the electrical properties with yet more experiments, and you put all that data together, and then you can kind of fudge it where you put the 3D models from one experiment with the electrical data from a different experiment and the chemical data from yet a different experiment. You put it all together and you end up with a model of a neuron that you can simulate in a computer. We have a supercomputer here. Hopefully someday that'll all happen on a single graphics card. Someday, but for now, it's a long story. Anyways, yeah, so that's kind of what this slide is trying to say. Oh, and right here we can see, once again, this is that column where if you just sort of took like a core sample of the brain, we have the top of the brain. This is layer 1, layer 6. There's not 6 layers here.
7.
Okay, then. But anyways, we can move on to the next slide. So yeah, the reason we build computer simulations, if it's not clear, is that then we can run the computer simulation and compare it to real data. And then we can validate that all this work is correct. And yeah. So this is a still image from one of those computer simulations. There's a brainwave happening, and you can see that like there's a dark part here that's not electrically excited, and then there's a part that's electrically excited around it. And and you can really tell that there's different layers going on. Like this layer here is like distinctly has different activity than like up there at the top. There's like not a whole lot going on at the top of sort of across the board. It tapers out. Yeah, this is the kind of stuff that, you know, we want to check that this matches reality. And once we have a good idea that this is what's really happening in the brain, then we don't need to keep cutting up animals and sticking electrodes into their brain, because that's really a pain in the butt and messy, and that's not my thing. I only do computer stuff. So this is sort of where I can take over, is once they have a model like this. We can move on to the next slide. Yeah, it's a pretty picture. Anyways, I don't actually know what the next slide is, so when it loads in for me, rabid rats. Okay, so we got rabies, and we sequenced the DNA of rabies. And rabies, I'm not sure if you're familiar, but it travels throughout the nervous system. Once you get bitten at an area, it infects a nerve cell, and then it travels throughout the length of the axon. That's the long bit of the neuron that connects it from the brain to the muscles. And it travels from your body all the way up into your brain, and it travels through the neurons and through the synapses. And we took just the mechanism that lets it travel through neurons and combined it with some tracer molecules that are fluorescent and light up funny colors. It's not lethal to the animal, I don't believe. You really don't want to get it into you, but I don't think it's lethal anymore, and it only jumps across a single synapse. Instead of infecting your whole brain, it jumps across one synaptic connection and then lights up the postsynaptic neuron, or actually presynaptic neuron, because it travels backwards through the neuron, through the system. Anyways, we can use this to trace the synaptic connections. The data is really pretty. We'll move on to the next slide. And we'll see more pictures of this. You'll notice there's two colors. That's because you get two data samples, two injection points in a brain. So if you want to like take a whole catalog of the whole brain, like by measuring every little area of it, you're going to need a lot of mice, and because you only get like 2 or 3 data points depending on how many colors you have to work with. So they went through a lot of mice to get these nice, neat wiring diagrams. But you take a lot of these samples of data, you align them all. We have areas of the cortex, areas of the thalamus, and they're like finding the connections between these two organs. And these aren't just like how do I say this? These are synaptic connections. These are actual functional connections in the brain, as opposed to anatomical connections where two things simply pass close by each other. So this is a nice piece of data to have. I think we can move on to the next slide. Yeah. It's loading. Anyways. Oh, install. Okay, we're almost done. So the debug port. Animals don't have a debug port. Evolution didn't make a debug port in the brain. But we can make animals do whatever we want. We have genetic editing. So a while ago, we figured out how to take 2 proteins from nature. We found the gene sequence that creates those proteins. And one protein detects calcium ions, which are an important signaling molecule in the brain. Whenever those neurons emit pulses of electricity, very often they'll also emit calcium within the cell. The cell will flood with calcium if the cell activates a bunch. And then the molecule lights up fluorescent green, and that's from a jellyfish. And we took these 2 pieces of DNA, stuck them together, did a bit of tweaking, put it in animal brains, and we're going to look at a video of that in a minute. But first, we're going to go on to the next slide, and then immediately after to the next slide. So 2 slides? Should say decoding the data. Yeah, so we use CRISPR to make these neurons in the brain. They light up. We put a camera in the animal's brain, surgically installed. And then we also can figure out what the data means. Yeah, here we go. The images on the top are what we showed the mouse, and the images on the bottom, we read the mind of the mouse because we installed a debug port. We're looking at its brain activity. And if we look at the visual cortex, that's the part of the brain that processes vision, we can, what you do is you start by showing the mouse test images, and you correlate the test images with the brain, the neural activity, and then you can sort of figure it out from there. It's not that simple as installing a camera. It's not like there's a little TV inside their visual cortex that we can just stand over their shoulder and watch. But yeah, and the final slide, and then we're gonna watch a video of this in action. But there should be one more slide. Yeah, and it's gonna say we have the technology.
Technology.
We have the technology to understand the brain. I've been gushing about all the data that we have and how cool it is and how high resolution it is, but really, we do have. I haven't shown you the data that doesn't look cool. There's a lot of data that I actually cut out of this because I wanted to keep down the time. Because there's going to be another part afterwards where I explain how the brain works. But yeah, we have a lot of data. We can take a neuron and we can find all the chemistry going on inside it. We can figure out all the electrical properties. We know what it's shaped like. Name a fact about the brain, an aspect that you might want to measure, and we can measure it. So this is my sales pitch as to why I think it's an interesting thing to study. And now we'll move on to those two videos. The next video in the This is attainable. When I was a kid, this was not attainable. We didn't used to have this technology. We didn't used to have CRISPR, but yeah, we're gonna get there. So we're gonna look at the mouse. Yes, here we go. Here's a mouse. When the video plays, it's alive and awake. It's interacting with its environment. It's conscious. We can read its thoughts, so to speak. It's living its life. And then over here, we have the data here is the raw video data at the top. And at the bottom, they've increased the contrast and stuff to clean it up and make it more visible where the neurons are and what they're doing. Yeah, so we're reading its brain activity as it's alive and awake, which if you had some theory about how consciousness worked or how the brain worked, this is the kind of data you'd want to test your theory against. Like, we can put the mouse in a maze and make them look for cheese. And after this video finishes, the next is going to be showing you how to decode the data, because I have a little video clip showing the mouse. Yeah. So.
Cool plot.
Doing a good job.
Oh, is it a load with the? Yeah, my video player is a little laggy. Yeah. So what we're looking at here, there's two types of data. There's the neuropixels, which is an electrode that was put into the brain, and then we have the 2p excitatory neurons. That's 2-photon imagery, and that's the GCamp calcium imagery that I was talking about. I believe is the same thing as the 2-photon excitatory neurons. So what we were looking at with the mouse in the last video is at the bottom here, and this is just an old-fashioned just wire stuck into the brain. And what you'll notice is that when you stick a wire into the brain, the data comes out with a lot of these little blips that are really rapid, because it has really good time resolution, whereas the imagery from the last movie had really bad time resolution, and you can see that here, where like a neuron will light up and it will remain active for a while. If you're not, I should explain what we're looking at here a bit better. Each of these lines of data is a neuron that we recorded from. And then here we have what we think the mouse was looking at. And over here, I don't know. This is the kind of stuff that you put into your science so that you seem... I'm just jealous of the visualizations. Here I am like writing on a whiteboard. Anyways, so...
Plot lines go backwards now.
Yeah. This is really cool data. We have tons of data. We can stick electrodes into mouse brains. And yeah. We can move on to the final video. It's going to be a lecture where I explained those algorithms in the brain. And then we'll do some Q&A. All right.
Think about a smart question.
How is information encoded into action potentials, and how do the dendrites decode that information and make use of it? Okay. Our journey starts in the 1950s and 60s when a pair of researchers by the names of Hubel and Wiesel stuck a bunch of electrodes into a cat's brain. And at the same time, they forced the cat to look at some images, actually short video clips of scenes. They were searching for correlations between what the cat is currently looking at and what is happening inside of the cat's visual cortex. And what they found was that neurons in the visual cortex emit action potentials in response to the cat seeing things. Hubel and Wiesel discovered that the purpose of neurons is to detect things. They won the Nobel Prize for their seminal work, and their experiments have been replicated many times and expanded upon. These neurons are constantly watching over the cat's visual inputs and waiting until the cat sees the object that the neuron is supposed to detect. And then the neuron emits an action potential, which travels throughout the neuron's axonal arbor to the rest of the brain, and that signals to the rest of the brain that the cat is looking at that particular object. Interestingly, they also found that when they showed things to the cat, most of the neurons did not respond by emitting action potentials. Typically, only a few % of the neurons in the cortex are emitting action potentials at any one moment in time. This makes sense when you consider that out of all of the things which you could possibly see, you're only seeing a few of them at any one time. And so only a few of the neurons are active. By the way, I'm going to use the term neuron activity to mean that the neuron emits an action potential, and a moment in time in this context is typically about 100 milliseconds, since that's how long it takes for the human visual system to consciously register a visual stimulus. So now let's start to analyze this system. We've established that neurons in the visual cortex detect objects, and then they transmit that information to the rest of the brain using action potentials. So in theory, if we could see the voltage of every axon leaving the visual cortex, then we should be able to decipher what the cat is currently looking at, or at least what the cat thinks it is looking at. But in order to extract that information from the neural activity, we need to understand how that information is embedded into the neural activity. How does neural activity represent information? Okay, so naively, we might expect each neuron to detect one object. And then there would be a one-to-one correspondence between the objects that your brain can detect and the neurons that detect them. This would make it very easy for other areas of the brain to decode the signals that are being sent to them. Each neuron would have a single very specific meaning, and each neuron could specialize in detecting things very reliably. This hypothesis is known as the grandmother cell hypothesis. Under this hypothesis, there should be a cell in your brain that recognizes your grandmother's face, and her face, and only her face. And there only needs to be one such cell. At first, this seems like it would be a good arrangement, but there are a few problems with this hypothesis. There are flaws with this encoding scheme, and we do not see these same flaws in real animals, and so we can conclude that this is not how real animals work. The most obvious problem with the grandmother cell hypothesis is that the maximum number of objects that you would be able to recognize would be equal to the number of neurons in your brain, because there's a one-to-one correspondence between those two things. However, we know that people stop growing new neurons very early in their childhood development, and yet you can continue to learn new things throughout your entire lifetime. Where would the neurons come from to learn those new things? Could it be that you were born with extra neurons that are reserved for later in life? No, scientists have ruled out that possibility. There is no such pool of reserved neurons. All of your neurons are being used throughout your entire lifetime, and yet you're able to keep learning new things. Clearly, neurons that have already learned to detect objects are able to learn to detect new objects. This implies that neurons are able to detect multiple different objects. Another problem with the grandmother cell hypothesis is that brain cells routinely die. Some people trip and fall and hit their head really hard, and that kills neurons. But even without concussions, neurons die of natural causes every day. Living cells are fragile, and all successful animals have evolved ways to cope with random cell death. Under the grandmother cell hypothesis, when those cells that recognize your grandmother die, you would lose the ability to recognize your grandmother. We know that this does not happen in real people. Healthy people don't just forget what their grandmother looks like, even if they get hit on their head really hard. Don't get me wrong, though, concussions are really bad for your brain, but they don't cause you to completely forget specific facts just like that. So clearly, our naive way of encoding information is wrong. There can't be a one-to-one correspondence between neurons and the objects that they detect. Each object needs to be detected by multiple neurons in case some of them die, and each neuron needs to be able to detect multiple objects, or else you won't have enough brain cells to detect everything that you need to detect. So we've disproved one hypothesis, and we've learned some new constraints on how the brain can encode information. The next waypoint on our journey is the dendrite. Before we can understand how neural activity encodes information, we need to understand how other neurons decode that information. Action potentials travel through the axons, shown in purple here, to the synapses, and then at the synapses they cause something called an excitatory postsynaptic potential on the dendrite, or EPSP for short. An EPSP is a brief pulse of electric current, and that raises the voltage of the dendrite by a little bit. When several EPSPs happen at the same time on the same dendrite, their electric currents add up, and together they have a proportionally larger effect on the voltage of the dendrite. Neuroscientists have found that dendrites have some interesting properties. Dendrites are normally not very conductive, meaning that when an EPSP happens on a dendrite, they don't really do anything. The electricity goes nowhere and it quickly leaks away. However, dendrites are full of a special type of ion channel that is voltage activated, and this special ion channel activates, it makes the dendrite much more conductive, which allows the EPSPs to affect the soma and possibly even cause the neuron to emit an action potential. This phenomenon is known as the dendritic nonlinearity, and sometimes dendrites are described as active dendrites. Normally, the EPSPs sum in a linear fashion, as shown on this portion of the graph. But when enough EPSPs happen together on the same dendrite, their combined effect is greater than the sum of their individual effects because of the special ion channels. That's shown on this portion of the graph up here. And getting back to our quest to understand information processing in the brain, this dendritic nonlinearity implements a sort of threshold. Typically, this threshold is at around about 10 active synapses. This is, in fact, another piece of evidence against the grandmother cell hypothesis. Now let's ask, why does the dendritic threshold exist? What is its purpose? What does it do? What problem does this solve? We've actually already touched upon this. The answer is that neurons can represent multiple objects. When a neuron activates, it's a signal to the rest of the brain that the neuron has detected one of the many different objects that the neuron is supposed to detect. And that information alone is very ambiguous, and so it's pretty much useless to the rest of the brain. This is why the dendritic threshold exists, to filter out that information. On the other hand, when a dozen neurons activate, and they all have one object in common that they all represent, then it's very likely that that's the actual object that the neurons detected. Let's look at an example of this in action. Okay, here is a chart representing a dozen different neurons. Each box shows the objects that the neuron is supposed to detect. This chart should really have about 1 million more boxes on it, but let's just focus for now on the dozen that I've shown. So, for example, this neuron would represent a house, a tree, or an apple. And this box would represent a neuron that detects a hammer, an apple, or a dog. And this box represents a neuron that detects an apple, a banana, or a fork. So let's suppose that all 12 of these neurons activated all at the same time and no other neurons activated. What do you think it would mean? Notice that all 12 of these neurons represent apple, and apple is the only thing in common that they all represent. So when we see an apple, all 12 of these neurons will activate and send EPSPs downstream to the dendrites that care about apples. The dendrites that care about apples are wired up to these 12 neurons. And when we see a banana, those same dendrites will receive 3 EPSPs from the 3 neurons here that also represent bananas. This is the purpose of the dendritic threshold, to differentiate between these two cases, to respond to the true signal and to filter out the noise. There are always going to be a few neurons that, by coincidence, represent both apples and bananas, but the intersection between those two sets of neurons is much smaller than the dendritic threshold, and so activating one set of neurons doesn't accidentally signal that the other object was detected. Okay, now that we understand the basic concepts, it is time to mathematically analyze them. But don't worry, I'm going to keep things simple. First, I need to introduce some new terminology. A sparse distributed representation, or SDR for short, is a vector of Boolean values. This is a list of ones and zeros. Each value in the vector is supposed to represent a neuron. If the neuron emitted an action potential, then it's represented by a 1, and if the neuron was silent, then it's represented by a value of 0. At every moment in time, your brain is generating a new SDR to represent its current activity. Right now, your visual cortex is outputting an SDR, and it represents the objects that you're currently looking at. Formally speaking, an SDR is a vector of n many bits where a many of the bits are 1s and the rest of the bits are 0s. The sparsity of the vector is of course the fraction of the bits that are 1s, and we're going to assume that the sparsity is always going to be a constant and predetermined value throughout all of our equations, because this assumption simplifies everything. Now let's define the dendrites and the synapses. Each dendrite samples from some of the bits in the SDR, and this sampling process represents the synapses on the dendrite. When the dendrite samples from a 1, that represents an EPSP happening to the dendrite. And if the dendrite receives more EPSPs than the dendritic threshold, then it's going to activate, which is a signal to the rest of the brain that the dendrite detected the thing that it's supposed to detect. And there is a whole process for deciding exactly which bits that the dendrite should sample from, but that's a whole nother story for another day. Great, let's analyze some properties of SDRs. I have an example here for us to work through. In our example, we have 2,000 neurons, 2,000 bits in our SDR. 40 of those neurons are active at any moment in time. Those are the 40 ones in our SDR. Each dendrite has 20 synapses, and the dendritic threshold is represented by the symbol here theta, and that is 12 EPSPs. First, let's count how many unique SDRs exist for any given n and a. The answer is n choose a, where choose is the mathematical operator for counting the number of different ways to select a many different combinations of n elements. This is another way to write the choose operator, and these exclamation marks here are factorial are the factorial operator. For our example here, this value is about 10 to the 84th, which is an absurdly large value. For context, there are also about this many atoms in the visible universe. There's actually about This is actually about 100 times larger than the size of the visible universe, but I mean, who's counting? When you get to things this large, who's really counting? And I just want to stress that this is a small example, that the brain has millions and millions and millions of neurons, and that we're dealing with 2,000 neurons. As you get more neurons, this equation simply grows larger. I mean, we only have 2,000 neurons, and we're already in the realm of larger than the visible universe. If you have millions of neurons, I mean, these representations are basically infinite. Now, not all of those SDRs are actually going to be distinct enough for the dendrites to tell them apart from each other, because if two SDRs have more active bits in common with each other than the dendritic threshold, then the dendrites are liable to confuse the two SDRs, and the dendrite will activate in response to both of those two SDRs, and then we consider them to be confused with each other. They're not distinct enough for the system to tell them apart then. In practice, though, this is not really an issue because there are just so many possible SDRs that they usually just don't interfere with each other like this. And this brings us to the next important property of SDRs, and that is that randomly generated SDRs are almost always distinct from each other. The probability that two randomly selected SDRs will have more active bits in common than the dendritic threshold is written here on the board. And for our example, this probability of two SDRs getting confused with each other is less than 1 in 100 billion. Intuitively, if you generate a random SDR with 2% sparsity, then each bit of the SDR has a 2% chance of being activated. So when we generate a second random SDR, and we're concerned that we might accidentally activate a bit that's already been activated in the first SDR a second time, well, there's about a 2% chance of that happening for each bit that we activate. So if you take a 2% risk 40 times in a row, chances are that you will lose that gamble at least once, and you will accidentally activate a bit that's already been activated. But what are the odds that you lose that low-risk gamble a dozen times out of 40 rounds of gambling? I say 12 times because in our example, that's the dendritic threshold. This property is what allows us to learn new things using old neurons that have already learned a bunch of other stuff. We can select a group of neurons at random to represent a new thing that we want to learn about, and we won't accidentally step on a group of neurons that already represents a different thing. We can randomly generate these vectors all day, every day, for the rest of our lives, and we will basically never collide with an SDR that we've already previously generated. The next important property of SDRs is that they're robust in the face of random cell death. Our example already has some nice big safety factors built into it, which protect us against random cell death, and our example isn't even that large of an example. As these systems get larger with more neurons and more synapses, they become more resistant to these sorts of random cell deaths. In our example, each dendrite has 20 synapses, but dendrites really only need 12 EPSPs to meet the dendritic threshold. And so we could lose 8 presynaptic cells and still guarantee that all of our dendrites are able to work correctly. But even if we lose more than 8 cells, as long as the cells that die are random and they're not correlated with anything in particular, then chances are that no one SDR that we care about will lose more than a few of its active cells. The intuition here is the same as to why random SDRs rarely interfere with each other. It's very unlikely that random cell death will destroy all of the neurons that represent any one specific object. In our example, if 1% of the neurons died, that's 20 of them, then it could in theory disable one of our dendrites. But the probability of that actually happening in practice is less than 1 in a trillion. So the final important property of SDRs is that they can be combined together. In the same way that algebraic sets can be unioned together, SDRs can be unioned together as well. Remember that two random SDRs will rarely interfere with each other? One SDR will not accidentally be mistaken for another? Well, we can actually activate two SDRs at the same time, and dendrites that are set up to recognize either one of them will correctly activate, but the other dendrites will not accidentally activate in response to the two SDRs that were activated. This property is useful for representing ambiguous situations, like when you're not sure what you're looking at. So all of the neurons just signal all of the possibilities and hope that one of them is useful, even if clearly they can't all be right. For example, you might see some movement out of the corner of your eye, and it could look like either a shadow cast by a tree blowing in the wind, or it could be the stripes of a tiger. And so your visual cortex uses this union property to signal both possibilities at the same time. It's both a shadow and it's a tiger. And of course, the rest of your brain is going to ignore the shadow and respond to the possibility of a tiger. The key to making these unions work is that even after combining the cells from several SDRs, the resulting SDR must still be very sparse. If there are too many cells active at the same time, then it will start accidentally activating dendrites that it wasn't supposed to. And this puts a limit on the number of SDRs that can be unioned together. I like to visualize what the space of all possible SDRs looks like. I like to imagine SDRs as points on the surface of a very high-dimensional sphere. This sphere is centered on the origin, and because our dimensions are Boolean values, the sphere is confined to the positive quadrant. Distances are, of course, measured using the Hamming distance. The radius of the sphere is the number of active bits in the SDR, which is usually quite sparse, meaning that all of the points on the sphere are actually fairly close to each other, and they're also pretty close to the origin of the sphere. However, the surface area of the sphere is astronomically large because surface area increases exponentially with respect to the number of dimensions in our vector space. Now, imagine that you're standing on the surface of this sphere and you look around yourself in a circle. How many different directions could you travel in? The answer is the number of different dimensions in our vector space. And so in our example SDR with 2,000 neurons, or 2,000 dimensions, we would be able to travel in about 2,000 different directions. This allows us to move around without running into anything that already lives on the surface of this sphere. Imagine playing the game Snake on the surface of this sphere. You would never run yourself into a dead end that you couldn't escape from. There would always be another direction you could turn towards to escape. Essentially, you can take any mathematical manifold and map it onto the surface of this high-dimensional sphere. No matter how complex your manifold is, it will basically always fit onto the surface of this sphere, and with lots of extra room to spare for expanding the manifold in the future. And what synapses and dendrites do is they determine if the current SDR is located on a particular patch of the surface of the sphere. They do this by sampling from the dimensions that point towards the sampled patch of the surface area and discarding the rest of the dimensions that are orthogonal to the sampled area. This projects the sphere from a high-dimensional space to a much, much lower number of dimensions, like 1. And all of the discarded dimensions collapse into the origin, and the remaining dimensions get summed together to point in a single direction, and then the dendritic threshold can differentiate between SDRs which are on or near the sampled area and the SDRs which got projected into the origin.
Thanks, neuroscience.
We can use this technique of subsampling and thresholding to isolate any patch of the surface area that we want to, if that makes any sense.
All right, still playing for me actually because
Anyways, I hope you've learned something interesting.
Yeah.
And so until next time,
Meow. That's all.
Yeah,
Excellent.
that was my presentation. I was really excited to learn about that back in the day because
math, and we can analyze the brain with math, and that's pretty exciting. Yeah. So I guess it's time for Q&A.
Clap,
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 clap, clap,
clap,
clap, clap, clap, clap. Yeah, I'm sure you have questions. Oh, yeah.
Hey, Frostnip, thanks for the
Hello?
thanks for the presentation. Two questions. One, you mentioned earlier, and I might have missed it, but how do we categorize neurons or synapses? I mean,
Neurons. Okay, so the way we categorize them is something called unbiased clustering. At least that's what I was talking about in the presentation. And so what you do is you have a neuron here. I'm going to get some pens out and draw some stuff. You have a neuron. I'm going to draw a cell body. It's a triangle. And we've got a bunch of dendrites. And we can measure like the length here. And we can measure like these angles. And we can do this across the entire dendrite and axonal arbor. We can do this for the whole neuron. We can measure all these little things. And then we can like do statistics on all those measurements to create like just sort of random measurements of the shape, like things like how much these things wind around, and then we can do a profile of how much they wind around versus distance from the cell body, and you get sort of these sort of profiles of like it's very windy here, but then it sort of straightens out here, and then we have just a bunch of data like this. It's not terribly meaningful, but it's meaningful to a computer that runs a clustering algorithm that can say, hey, some neurons have this profile and some neurons have a different profile. And then you can sort of start to automatically generate categories of neurons.
gotcha. And do these categories affect the function of the neuron, or is it just
Yes.
the shape? Got it.
Well, those are two things, and they're also the same thing. The function of the neuron is a property of the shape of the neuron, as well as the chemistry on the surface of the neuron, all the things that are embedded in the surface that like move ions in and out of the neuron, all that electric stuff, and there's a bunch of chemical stuff. We also stick a needle into the cell body and suck out the nucleus of the cell where the DNA is, and then we can read out which bits of DNA are being expressed and creating proteins. That's just a different data point, and it gives us different information about the classification of neurons. We get mostly they correlate with the shape. The shape mostly correlates with everything else, but yeah.
Gotcha. Thank you for that. So the second question is in terms of SDRs. So in order to recognize something, right, you have to have some threshold of neurons responding. Is the occurrence of neural activity, neural responses, is it distributed relatively normally across neurons? So like neurons respond to roughly the same number of inputs, but to different ones, or do some neurons respond much more frequently to others? So like in terms of like say language, for example, right? Go ahead.
Most neurons have fairly regular activation rates, especially if you measure them over a long enough period of time. The brain tries to keep everything fairly normal. You don't want to have one cell that's twice as active as other cells. Part of the reason we have these distributed representations is so that we don't end up with one cell that's representing the really important thing and the other cells aren't representing anything important. They all represent different things and many different things, and so on average, they all have relatively equal, I guess. If that answers, there's a bunch of homeostatic mechanisms involved.
Do we know what induces that behavior? Like what quote unquote evenly distributes the activations of neurons?
Kind of. There's chemical mechanisms embedded within the neuron that limit how active it can get. And there's also inhibitory mechanisms. We do kind of know those mechanisms, yes.
Okay.
Yeah.
Gotcha. Yeah, I don't want to hog Q&A, so I'll talk to you after.
I think the key word you're looking for is probably homeostatic feedback in the brain. It's everywhere. Anyways.
Got it. Thank you.
Corliss?
Oh, there we go. Hey, thank you for the presentation.
Thank you for listening.
This subject is hella fascinating to me. I really love cell physiology, but I am curious on the computational side that we're capable of measuring EPSPs, but can we do IPSPs as well as was it graphing neural backpropagation?
So IPSPs, we can measure EPSPs
What are
with
IPSPs
electrodes.
also?
Those are inhibitory postsynaptic potentials. So
Gotcha.
you have a synapse where you've got like some axon, and the axon has like a bulge in it with a bunch of chemicals, and this will release neurotransmitters. And there's those synaptic spines that we were looking at earlier, and they sort of are attached to a dendrite, but also they sort of go towards here. And basically, it's just a different chemical reaction here. And we do we do understand them. They're a bit less studied. They're a bit less exciting than the EPSPs because EPSPs transmit data in the cortex, and these are inhibiting. They do a different thing. We do know a lot about them though, especially nowadays. I feel like we didn't always know as much about IPSPs. What was the other question you had besides IPSP?
It was, are we also capable algorithmically to measure neural backpropagation?
Oh yes, and I'm not sure neural backpropagation means what you think it means. So there's neurons, and they have like dendrites that are far away, and you have the cell body down here, and this is where the action potential is initiated, and it's like this pulse of electricity, and it propagates back throughout the axons. And if that's what you mean by backpropagation, then yes, we can simulate the flow of electricity through neurons. So yeah, but this is not the backpropagation algorithm from deep learning.
Okay.
It's called the same thing. That's sort of a source of confusion, but yeah.
Thank you.
Oh, okay.
Yes, thank you for the presentation. Really interesting. In particular, I was quite surprised to see the multiple dimension system on the sphere. It's exactly how the clip models in machine learning work. And all of that, I do see it working. Like I see we have all of the building blocks to make a Turing complete machine with neurons. But something I'm still missing and still cannot wrap my head around is how does the actual learning work? Because we see it's possible to create a machine with neurons capable of computing anything, but I still fail to see how the feedback mechanism work. How does the conscious part of the brain realize that something was good and reinforce all of the neurons that led to that result? Because you need quite a complex architecture. Like in machine learning, doing a feed forward is really easy. The backpropagation is the hard part. And I talked a lot with friends which work in the domain, and I don't see any of that being really figured out yet.
So what I would say is there's a lot of learning mechanisms, but they all happen at the synapse. So Hebbian learning happens at a local synapse. There isn't it's just the physiology of the brain doesn't permit things that are global. I mean, there's some things that are global. There are some transmitters that transmit very quickly across the entire brain, like nitrous oxide is normally in your brain in small levels, and fluctuations in it transmit very quickly across, like, globally the whole brain. But what you're looking for is like the reinforcement learning mechanisms with like a reward signal that propagates from the dopamine centers, and those are not a single cohesive signal. There's a bunch of neurons that transmit it all over the brain, and when you look at the chemistry, it appears to all be happening at the synapses locally. So we do have a lot of mechanisms, but they don't always... We know a lot about how the brain learns, but it doesn't always mesh with how we think the brain learns and how AI systems work. The AI systems in the brain don't really work in the same way. There's no backpropagation. I mean, there's backpropagation within the neuron, but it's not like a global error signal that backpropagates.
There need to be some kind of feedback mechanism, in particular for very complex tasks. Like a human body is capable of
For
human brain is
reinforcement
capable
learning,
of
yes.
understanding consequences on quite a long term. You can learn that something was bad even though you did it 2 weeks ago, and so that's incredibly complex.
Well, that 2 weeks ago, if you want to learn over 2 weeks, you're going to need a long-term memory. You're going to need to remember it at the day, and then that's in the human brain that actually gets shuffled from part to part. That memories start out in your hippocampus, and then they get transferred to your cortex, and then later you might like think about this event that happened, and now you're consciously recalling these two things, and now you're putting two and two together. That level of like thinking is pretty in depth and unique to humans. And yeah, that's harder to research. There is some interesting research into how consciousness works. It's something I've looked into. It's a bit beyond the scope of the Q&A, though. But it basically revolves around, at least what I've seen the research revolve around, is a thing called conscious access, which is this idea that you can decide to think about something and then you think about it. Like if you just think about the thing that's red and it pops into your vision, you're like, oh yeah, this. Or like think about something that's like round or, and it just pops into your vision. It was always there, but you weren't consciously aware of it until your auditory cortex like told your visual cortex like, hey, look for the thing. And there's a whole process for that. We actually have some ideas how that happens. But that's sort of, I think, where the state of the art of the field is.
It makes a lot of sense for dreams to exist for long-term learning in this case, because you are able to refire the same neuron that led to the scenario, and you already know the output, so you can already do the backpropagation of an event while you know the output like 2 weeks later already. So quite interesting. Thank you.
Well, what I would say is you see how it works. It probably doesn't use backpropagation though. But you do have that recall mechanism.
Yes. It is very biological. It's not a proper mathematical equation.
Yeah. Satala?
Yee! Thank you so much for the presentation. It was really interesting. Now, I know earlier we talked about the different areas of the brain and how they practically function. A lot of that research is being done through MRI. And of course, the neuroscience that what we've learned from MRI is now feeding into psychological studies, and you yourself already kind of showed a little example of how that's really helpful. I'm curious, if we were able to map the entire brain, neuron by neuron, and able to understand how neurons work, how do you feel that would excel the research in understanding psychology or understanding how the brain operates in a more meaningful way for people who are not necessarily research scientists?
It would certainly be helpful for medicine. There's a lot of things we don't understand about the brain. What I will say is I think we're going to understand the brain in terms of general principles and like the computational stuff that I do. I think we're going to understand the brain before we understand the human brain medically. The human brain is, medically speaking, more complicated than the mathematical construct of the brain that like I simulate in a computer. So there's just so many different things that can, I don't want to say even go wrong, just be different from person to person. It's going to be very helpful to have an understanding of the brain for medicine, though. There's a lot of things that we don't have. Medicine is a challenging field. I think I'm just going to leave it at that. I honestly don't study medicine except if I can understand the computations involved. I mean, you find studies where people have some part of the brain that got damaged and then they lost some ability, and then we can sort of infer some stuff from that. But beyond that, I don't really study diseases.
So maybe to put it a little bit more in your field then, do you believe that we would be able to actually produce a functioning artificial brain? And if we were, do you think that would help us better understand some of the psychology of how it operates? Or do you feel like this would be
that particular
maybe
brain,
useful
yes.
for other
What I would
purposes?
say is if you want to talk about recreating the brain and putting it in a jar and harnessing it to do useful work, you're going to be looking at things like monkey brains, where, you know, a trained monkey can work in a factory and operate a mechanical arm as though it was a real arm, or balance on two legs. But that's not a big brained human activity. That's like something that mice can do. I mean, mice don't have the legs to do it, but they have this. They have all the other stuff going on in their brain that if all they wanted to do was stand on two legs and like reach up to something, it's like small four-legged animals can do that. And that would be like really useful, is if all those robots that we have the chat bots, we have the calculators, we have the big brain stuff, but now we need the legs and the arms and the motor cortex and the visual cortex so that self-driving cars don't run into stuff, you know? And these are, I don't, what was your question again? Sorry.
Effectively, if we had a computer model of a brain that was functioning, and we based it off of the way the human brain works, what would you feel the advances would be? And would we, like, I guess personally, because I do a lot of anthropological type research, speaking with a lot of people, and there's a lot that we don't know about how people operate, and there's a lot that we would like to try and figure out and learn more. I mean, is that a field that you think could be a benefit, or are there other types of fields that you think?
What I would say is that the human half of the neuroscience is going to be more difficult than the mouse half. We're going to understand how a mouse brains work before we understand how a human brain works. And for the purpose of medicine, that's important, because mouse brains are much less medically interesting. And half the people working in neuroscience are only there for medicine, and the other half are there because they think it's a neat thing to study and they want to understand how mouse brains work. And some people, there's a bit of a divide there. I feel like a lot of the money coming into the field is obviously coming from the medical side of the field, where they want, you know, there's real investment into medicine. But yeah, I think there are going to be advances. It's just going to be a lot more difficult for what you're looking for.
Thank you so much.
Hello. It was very interesting. I mean, it was amazing what you did there. I have a question. I just don't understand one thing. What are the building blocks, or what actually structures all the neurons and says, hey, this part of the brain is responsible for that, and that part is responsible for that? What actually says that? Like, how is this actually decided?
Yes. When you're born, all of the neurons swim to specific parts, and there's a bunch of like chemical messages that they communicate to each other to say, hey, I'm this part of the brain, and the other one says, oh, I'm that part of the brain, and then they all talk to each other and coordinate as to where they need to be. This is a field called EVO-DEVO, evolutionary development. If you want to look something up in a book or on Wikipedia, they'll have lots of pictures of the brain being developed. And what you'll see is that
you have also the different like you have all the different neurons, so that's all decided by like inputs, or how I don't understand. Sorry. Just continue.
Okay, so to go to like the root of your question, a guy named Turing actually figured out that you can basically make a chemical gradient where you have a little bit of a chemical on one side and a lot of chemical on the other, and then you have some chemical reaction, and you have a bunch of cells that are going to live here. And you have some chemical reaction that only responds when the level of the chemical your hormone gets above a certain level, and so you end up with like this becomes like a leg on like a fly, and this becomes like the torso because the torso had more of some chemical, and this chemical is emitted from over here, and it like diffuses out, and as it diffuses, it gets less and less, and this is the chemical mechanism. There's sort of chemical little finite state machines and little diffusion mechanisms to try and make patterns. And once you can make a pattern like a zebra stripe pattern, you can sort of start, or like a French flag pattern was another one, where you can like make patterns out of cells using chemical gradients. And that's just the basis of it, of the patterning.
So basically like a symmetrical foundation, like in physics, in a sense, like cinematics inside pipe functions. Okay, but one thing that also I don't know how to frame it to let it understand. So you tell me in the video is like, oh, cell size and you know the grammar hypothesis or whatever, right? You said about that, and it makes no sense because if this dies, then you lose the information, and you have all these neurons that are responsible for that. So if they're so diverse, right, and you have all this diversity, then it's weird, right? And they adapt all the time and change. In some cases, you know, there's neuroplasticity changes. Then I cannot understand how the fuck this works. It's hard to understand for me. Like
it is complicated. What I would say is that we understand there's different levels of understanding, and many neuroscientists themselves don't understand it all. They pick a level of specialization, and some people specialize in like the cellular chemistry and they run a bunch of chemical experiments. Some people study like the connections between different brain regions and they use those rabies tracers, and they don't have to worry about the electric pulses going through the neurons. They just map out the wiring diagram. So it is a overwhelmingly complex thing. I'll admit there's a lot of stuff I don't understand.
Understood. Now there's a second question that
theories.
I have. The second question is: so let's say you could, you can use CRISPR, right? You use a virus, and this virus infects the brain. And what it does is it creates like either it creates polymers, right, or metal carbonites. So it creates like it conserves the whole brain. So suddenly you conserve all the brain, all the things, and with a small electrical impulse you can like basically it becomes a fossil. With a small impulse you can retract where this impulse is going, what's it doing, basically by just having electric current put into the brain. Is this possible? Could you do stuff like that?
I've never seen anything like that. All of the methods I've seen are rather invasive. Involve cutting open brains.
No. You have a brain that's already you wanna conserve. You wanna conserve the brain, right? So you change the whole neural structures.
You can't run the neurons like if it was live.
Normally not, but what if you tell the cells change to metal carbonites or change to
that's
right?
not
polymers.
really gonna work. That's
or code or
the chemistry
virus
to do that's pretty challenging, and I think you'll find that the brain's already found a pretty efficient solution.
Yeah, but your aim is to gather all this data. So if the easiest way is to make it in a fossil that can be activated and shut on and off, basically.
Okay.
If we could do that, that'd be great, but we can't.
I think that's pretty far beyond our capabilities these days. There's a lot of interesting research into how to look at the brain under a microscope and to fix it into place, but yeah.
So, thank you very much. Thanks for.
I don't know if I answered your question, but yeah.
Mm-hmm.
Go for it. Yeah, I found Seth Ian's question very interesting, and I've read a little bit about what you're talking about, and I thought that was interesting. Yeah, it's similar to in plants with their roots. They have what they call an apical cap. It's every root at the end of it has a special chemical that's attracted to nutrients and stuff, so it affects the growth, like left and right. You know, the more you see something on the left, it'll make the right side grow more, so you bend left. And so in a similar way, we have I think it's sirtuins, or some of the chemicals that are used, that's as the brain is growing, the cells where it needs to go actually line a path. They're producing the right chemical so that as an axon grows, it will be attracted to those and be led along the path rather than driving itself when it's in the neuron. It's being given a path by the DNA that made sure that the right cells would express those paths all around. So they automatically wire themselves. That's one way I think of wording it.
That's really interesting to hear. You might know a bit more about that than even I do, to be honest. It's the one that's not my area of expertise. What I have seen, there's some really cool videos on the internet where you can find they're not cell caps. I don't really remember what they're called.
Yeah, that's what plants call them. Are called for
in
plants.
neuroscience, there's a different thing. It looks like you have like an axon or some neural protrusion, some little tentacle sticking out, and it will like grow, and as it grows it like waves around, and there's like little hairs that are constantly on it, on the tip that it sends out, that are like waving around, and it grows where this is, and it keeps going. This is a thing that we've seen under microscopes, is just the little hair on the tip that detects things.
That is
so neurons
exactly
have a similar thing.
It's exactly the way that roots do it. So it's a conserved feature amongst different species. And so I thought that was interesting, if that helps him in understanding it, viewing it that way. But another thing I had wondered about, though, is the inhibitory paths that, from what I've read on dendrites, that I've found out that fascinating. They're finding out how much each dendrite itself can have more than one function of excitatory and inhibitory, and functions by the other connections that are around it. And that they also morphologically change over time. The length of time that something is true, they said, I think if I remember it right, because I read this months ago, but that it will start as like a spike, but that if it continues to be true and useful and the thought about, and the neuron is being triggered over time, it'll round it out. And it's more permanent, whereas the spikes can be reduced by future activity or inactivity.
By spikes, you mean like action potentials, right?
Physical formations.
Physical formations. Oh, yeah. Okay. What I will say.
Knew about that, or could throw anything my way considering it.
I know a little bit about neurons growing. Neurons do grow. You do get new neurons throughout your lifetime. And you can also lose them. They come, if you have a dendrite, typically there's like 10 times as many excitatory inputs, and they come in on the spines. And then you have inhibitory inputs that just sort of come in on the dendrite, and the inhibitory ones there's about 10 times as many excitatory as inhibitory, just give or take, just roughly speaking. If you don't have any inhibitory neurons, then what happens is a bunch of excitatory neurons turn on. They turn on the dendrite turns on the neuron. This is a neuron, it emits more action potentials, which then go off and trigger more neurons. And if you don't have any inhibition, you get a runaway feedback loop, runaway positive feedback. So the brain is full of inhibition and inhibitory neurons that just sort of keep a lid on this, and they basically raise the threshold to activate and turn things off.
Is there anything that you've known, because this I haven't looked into, this is not one of my major topics, it's a side thing, but I love it, is that they can trace any other neurons that are downstream or another part, that when something is found to be not true, can act as one of the inhibitory ones on the original neuron to damper its effect to it?
I believe you, and I'd love to see the study where they figured out how to measure that. That sounds
That's
really
what I was
interesting.
wondering, if you knew anything more on that.
What you're saying is that you have an excitatory neuron, it's backwards, and this excitatory neuron sends an axon to an inhibitory neuron, which then, is that what you mean? And you want to know like where these connections are?
Somewhat like, it will go to the areas it's supposed to say, hey, this is true for processing. But if you had some time, either currently or later on, find out, oh, well, and you get to unlearn that now, or it's not true at all, then you must have some kind of activation that there's cells that by having some input to them, they send a signal and can inhibit that neuron, from then on, something that regulates it so that it downgrades that input that it was formerly getting from the other neurons. To say, oh, when these things happen, it's still not true.
Yeah. I'd love to read the study. That sounds kind of complicated. There's a lot of people who look at neurons and measure them and build these big 3D models, and then you populate them with chemicals and electricity, and then you get a bunch of them and you put them together in a circuit and you try and figure out what they're doing. And this sounds a bit like that, where they looked at a circuit and they had something they thought it should be able to do, and they're trying to like match what they think it should be doing with a model of reality. It sounds interesting. I don't know much about it though.
Okay. Yeah, no, I didn't read one particularly on that. I know of a study, but they say that there are inhibitory things. So I was wondering about a mechanism, if there's one known and has been mapped out in any way that doesn't.
That's a very specific function as well. And usually inhibition is just sort of, I don't want to say that that effect isn't happening because it could be. I don't have data on it. Inhibition is an interesting topic. I could go on about it.
I didn't expect you to. I was just throwing a Hail Mary, wondering on that. Okay.
It's sort of the other half of the brain. 90% of the brain is excitatory, and then there's 10% that's inhibitory that is like actually half of the functionality. And then someone brings up astrocytes, and it turns out half the cells in the brain aren't actually neurons. They're actually other cells that don't emit action potentials, but are full of chemicals, and they are actually electrically active, and they connect to neurons in all sorts of weird ways. And we don't really know much about them. It's sort of this other thing going on that we've just started to scratch the surface of.
Yeah. I know that they also, though, do have a structural support system, so the brain's not a bunch of mush, but holds it like a gel instead of fluid.
There's a phenomenon where you'll have like a synaptic connection, where you'll have sort of two things come close to each other, and then you'll have an astrocyte that sort of reaches out and builds a glob around the whole thing, and it sort of holds all of it together physically, and it also confines the chemicals so that the chemicals don't leak out. It makes it harder for the chemicals to leak out because there's a bunch of chemicals getting passed back and forth in between these two neurons. And so that's one thing that astrocytes do, is they support this chemically and physically, and they sort of control this leak effect. But there's all sorts of things they're doing clearly that who knows.
Very interesting. Yeah, I didn't know it did that. I know myelin is like, would that also be like between gaps in myelin sheath sections of cells?
Those are different astrocytes that maintain myelin sheaths. There's just a lot of little cells in the brain that keep things working. I'm gonna get a cup of water real quick. Just a sip.
Next, maybe? Yeah. Could we pass along questions?
Oh, okay.
Yeah. Don't worry, Frostnip.
Yeah, it's all right.
Okay.
Sure, no problem.
Thank you
I
for
was
the.
just going to say when you get in the water that I'd say I don't want to hog the whole thing. So good timing.
Okay. Yeah. I'll be around later.
So thank you for the presentation. It was very romantic. This is half question, half comment. I heard about a company recently called Final Spark that released it's called a neural platform, and they provide a cloud Python API to interface with human brain organoids on like a chip. That provides like live readings for you to stimulate them and stuff. I was wondering if you'd heard about this because I find it deeply unsettling.
I'd heard a little bit. I didn't look into it. People are always trying to get human brain organoids. Those are human neurons grown in a petri dish, and trying to get them to play Doom. We're going to build that infinite torment nexus. Yeah. I don't put a lot of weight behind those things. It's neat, but I'd rather look at a brain under a microscope or in a real animal, or even better, an actual computer simulation of it where I can do anything I want with it. But in a petri dish,
you have like random neuron types, not necessarily in a structure. It's really neat that you can take random neurons, throw them in a petri dish, and they will assemble into sensible things that appear to like process information and form structures and do things that brains appear to do. But if you look closely, you'll notice that it isn't actually a brain. All that Evo Devo stuff that the question was about earlier, where it has to form in the right layers and in the right places so that the neurons are positioned to make the right connections. There are very specific wiring diagrams where some neurons talk to others, but they don't all just talk to each other in a petri dish. So I think scientifically it's not the most useful thing. It is neat. Maybe someday we'll all have cars driven by little brains in petri dishes.
That would be awesome. Thank you. Okay. Yeah, I just wanted to ask because it was a curiosity.
That is a curious thing. Okay. All right. Thank you for listening.
Yay.
Meow meow.
Wonderful presentation, sir. Great job.
Thank
A wonderful presentation from Frostnip. Thank you very much for that. That was Frostnip's lecture on computational neuroscience. We will be moving to another post-lecture world in a few minutes. Thank you again for Frostnip, as well as our YouTube attendees. Have a great Saturday, everyone.